Hello, welcome to my journal club presentation. My name is Brittany Chang, and today I'll be presenting on a study titled Dietary Fiber Content in Clinical Ketogenic Diets Modifies the Gut Microbiome in Seizure Resistant Mice. I'll take you through the study, analyzing its research, objectives, methodology, findings, and broader implications. So, why does this research matter? Epilepsy affects over 50 million people worldwide, according to the World Health Organization, and for about 30% of patients, medication simply doesn't work, according to Loshare. That's why alternative treatments like the ketogenic diet have gained attention. But here's the problem: not everyone responds to the ketogenic diet the same way. And why is that? Well, that's what the study sets out to explore. Specifically, they asked, could dietary fiber play a key role in making the ketogenic diet more effective against seizures? They found that ketogenic diets with higher fiber content didn't just change the gut bacteria, they increased microbial diversity, and they turned out to be critical because more microbial diversity was linked to better seizure resistance. But not all fibers worked the same way. Certain types are more effective, and when they looked at why, they found they were linked to specific metabolic pathways which were associated with neuroprotection. So, what does this mean? Well, when we think of the ketogenic diet, we typically focus on its high fat, low carb content. But this research suggests that fiber is just as important. Without it, the ketogenic diet might not be as effective, or worse, it could actually increase seizure risk. These findings open the door for new strategies in epilepsy treatment. If we can fine tune fiber intake, we might be able to enhance seizure control, especially for people with drug resistant epilepsy. Now, what did the authors set to answer in this paper? The authors aimed to explore an important but under-examined question. How does dietary fiber influence the gut microbiome in seizure resistance in the context of ketogenic diets? While ketogenic diets have been widely studied for epilepsy, its interaction with the microbiome, and specifically the role of fiber, has not been. Their goal was to identify whether fiber could enhance ketogenic diet's efficacy by fostering beneficial gut microbial communities and metabolic pathways associated with seizure protection. So, what is epilepsy? Epilepsy is a chronic neurological condition characterized by recurrent seizures, which can significantly affect daily life and cognitive function, according to the World Health Organization. And now, while medication is the primary treatment for seizure management, nearly one-third of patients continue to experience seizures despite pharmacological intervention, which really highlights the need for alternative approaches, according to Loshare. And one approach is the ketogenic diet, a high fat, low carbohydrate diet that induces ketosis, which is a metabolic state in which fat is converted into ketones for energy instead of glucose. And this shift in metabolism has been shown to reduce seizure frequency in some epilepsy patients, especially those with drug resistant epilepsy, according to Neal. The ketogenic diet is particularly appealing because it offers a non pharmacological dietary intervention that can be controlled and also tailored to individual patients. And then on top of that, dietary modifications are often more accessible, cost effective, and non invasive compared to lifelong medication use, which really does make them an attractive option, especially for patients in resource limited settings. And this is emphasized by Pauli in his research as well. Beyond treating existing epilepsy, understanding the link between diet and seizure resistance may also have preventative implications. Individuals with high risk of developing epilepsy, such as those with family history of seizures or neurodevelopmental disorders, could potentially use dietary strategies to mitigate seizure onset. So, research into diet based interventions could help shape preventative health strategies that improve neurological outcomes in at risk populations. So, the ketogenic diet has been recognized as an effective dietary therapy for epilepsy for quite some time, particularly in cases where medication fails. And the ketogenic diet works by shifting the body's primary fuel source from glucose to ketones, which may reduce neuronal excitability and seizure occurrence. However, emerging evidence suggests that the gut microbiome also plays a key role in brain function. The microbiome gut brain axis is a key area of interest when it comes to epilepsy research because changes in gut bacteria can also impact neurotransmitter production, inflammation, and seizure susceptibility. Now, in the recent years, studies have shown that ketogenic diets truly change the gut microbiome in a profound manner, which enriches bacterial species that contribute to neuroprotective effects, according to Olson. Now, despite this growing body of research, one aspect that remains understudied is the role of dietary fiber and ketogenics' effect on the microbiome. Fiber is a key nutrient that supports the growth of beneficial gut bacteria, yet most ketogenic diets are extremely low in fiber due to their carbohydrate restrictions. So, the main research question in the study is whether dietary fiber and ketogenic diets affect the gut microbiome in a way that improves seizure resistance. Now, researchers aimed to determine if modifying fiber intake could enhance ketogenic diet's benefits by promoting beneficial bacterial populations associated with neurological health. The authors hypothesized that dietary fiber could enhance the effectiveness of ketogenic diets by selectively modulating the gut microbiome. They expected to observe an increase in beneficial bacteria associated with neuroprotection and an enrichment of microbial pathways related to metabolite production, inflammation control, and neurotransmitter synthesis. Now, how was this done? To better understand the link between dietary fiber, the gut microbiome, and seizure resistance, these researchers designed a study using different ketogenic diet formulations. Mice were placed on one of 5 diets: a standard ketogenic diet, a modified lower fat ketogenic diet, a medium chain triglyceride based ketogenic diet, a fiber supplemented ketogenic diet, and a standard control diet. These diets were administered for 7 days before the mice underwent seizure susceptibility testing and microbiome analysis. Now, to examine how these diets influence the gut microbiome, researchers collected fecal samples and analyzed them using Illumina Nova sequencing, which is a high throughput method that provides detailed insight into microbial communities. They used 16S rRNA sequencing to identify bacterial species and shotgun metagenomic sequencing to assess metabolic shifts in the microbiome. This approach allowed researchers to not only classify the bacterial species present, but also determine how dietary fiber impacted the microbiome's metabolic function. But identifying bacteria wasn't enough for this study. The study also explored how changes in the microbiome influence neurotransmitter biosynthesis and inflammation pathways, which are incredibly important for the seizure resistance. By linking dietary fiber intake to specific microbial and metabolic changes, these researchers hoped to uncover the mechanism behind the anti-seizure effects of fiber-rich ketogenic diets. So, in addition to taxonomic classification, metagenomic functional pathway analysis was performed in order to assess how dietary fiber influenced microbial metabolism. This technique identifies which biological pathways are enriched or depleted in fiber supplemented versus non supplemented diets, which provided insight into potential metabolic shifts that could influence neurological function. So, beyond microbiome analysis, the study also examined the effects of dietary fiber on seizure susceptibility. Now, to measure seizure resistance, researchers used a 6 Hz psychomotor seizure test, a widely used model for epilepsy research. And this test determines the electrical current required to induce a seizure, providing a direct measure of seizure threshold. Additionally, researchers recorded latency to exploration, which is basically it measures how long it takes for a mouse to resume normal activity after a seizure. This served as an important indicator of post-seizure recovery and neurological function. And to further understand the impact of dietary fiber on the gut microbiome, researchers conducted microbial diversity analysis. They assessed 2 key factors: alpha diversity, which measures the richness and the evenness of microbial species within each diet group, and this was done using the Kruskal-Wallis test. Meanwhile, beta diversity evaluates differences in microbial composition between diet groups, and this was measured using the Permanova statistical method. This accounted for dietary effects on microbial communities. Now, this approach allowed researchers to determine whether fiber supplementation truly altered microbial diversity and overall community structure. And at the end, to ensure statistical clarity, researchers applied ANOVA with Bonferroni correction when comparing seizure thresholds across different diet groups. This method minimizes the risk of false positive results by adjusting for multiple comparisons. And for microbial diversity assessments, the Kruskal-Wallis test and Permanova were used to evaluate differences between diet groups. So, by integrating these statistical methods in seizure testing and microbiome sequencing, the study ensured that observed effects were accurately measured and true to the study. Now, let's take a close look at this figure to see what it's telling us about how different ketogenic diet formulations affected seizure resistance in mice when exposed to 6 Hz seizures. So let's start with the first panel A to the left. We see the breakdown of macronutrient compositions in these different diet formulations. And the key difference is that the standard ketogenic diet with a 4:1 fat to carbohydrate and protein ratio and the medium chain triglyceride based ketogenic diet with a 2 to 5 to 1 ratio both have high fat ratios and contain a variety of dietary fibers, while the modified ketogenic diet with a 3:1 ratio severely lacks fiber. In panel B, it outlines the experiment where mice were placed on one of these diets for 7 days before undergoing seizure testing to measure how much electrical stimulation was needed to trigger a seizure. And in panel C is where mice on the standard ketogenic diet with a 4:1 ratio and the medium chain triglyceride based ketogenic diet with a 2.5 to 1 ratio had significantly higher seizure thresholds compared to those on the standard control diet. This meant that they were more resistant to seizures. The standard ketogenic diet with a 4:1 ratio in particular provided the strongest protection. But in addition to that, mice on the modified ketogenic diet with 3:1 ratio actually had a lower seizure threshold, which made them more susceptible to seizures. This suggested that it's not just the fat ratio. Dietary fiber could be a critical factor in seizure protection. And you might think, why does this matter? Well, when we think of the ketogenic diet for epilepsy treatment, we usually only focus on its high fat, low carbohydrate nature. But this study suggests that we also need to look beyond the fat content and also look at the fiber composition. The lack of fiber in the modified ketogenic diet with a 3:1 ratio appears to reduce seizure resistance. And this is likely because it altered the gut microbiome and disrupted key metabolic pathways. Meanwhile, the standard ketogenic diet with a 4:1 ratio and the medium chain triglyceride based ketogenic diet with a 2.5 to 1 ratio create a microbiome environment that enhances seizure protection. These findings could truly reshape how we approach ketogenic diet therapies and suggest that fine tuning dietary fiber might improve seizure control in the long run. Now, if we continue going forward in figure 2, we can take a look at this and see all about the microbial diversity. We see that the standard ketogenic diet with a 4:1 fat to carbohydrate and protein ratio and the medium chain triglyceride based ketogenic diet with a 2.5 to 1 ratio significantly increases the Shannon's diversity index and species richness compared to the standard control diet and the modified ketogenic diet with a 3:1 ratio. But what's really interesting is that the modified ketogenic diet with a 3:1 ratio, which lacks fiber, doesn't boost microbial diversity at all. In fact, it actually reduces species evenness, meaning that the gut microbiome in these mice is less balanced, which could be a bad sign for seizure protection. But if we go into panel B, we can get a closer look at the overall microbial community shift with different diets. And the key takeaway to take from that panel B is that the modified ketogenic diet with a 3:1 ratio clusters separately from the seizure protective diets, the standard ketogenic diet with a 4:1 ratio, and the medium chain triglyceride based keto diet with a 2.5 to 1 ratio. This tells us that the microbial composition in the modified ketogenic diet with a 3:1 ratio is fundamentally different, which likely means that it's missing key bacterial populations that help with seizure resistance. And in panel 3, we can look at the metabolic pathways influenced by these diets. The standard ketogenic diet and the medium chain triglyceride based ketogenic diet promote pathways involved in amino acid and nucleotide biosynthesis, which are beneficial for brain function. However, the modified ketogenic diet increases pathways linked to carbohydrate metabolism and fatty acid degradation, which suggests that it might be fueling a microbiome environment that does not support seizure resistance. This is truly emphasized by panels D and E, where the standard ketogenic diet and the medium chain triglyceride based ketogenic diet share key metagenomic features linked to seizure protection, things like reduced sucrose degradation and increased quercetin biosynthesis, which are connected to neuroprotection. However, The modified ketogenic diet did not show those same beneficial metabolic features. So, this shows that fiber content in a ketogenic diet plays a crucial role in shaping the gut microbiome, which in turn influences seizure susceptibility. Now, in this fourth analysis of this figure, we can take a look and see that we are looking at whether adding more fiber to an already fiber containing ketogenic diet can further enhance seizure resistance. Panel A lays out the experiment where mice are fed either the standard ketogenic diet or the same ketogenic diet supplemented with an extra fiber mix. This extra fiber mix included inulin, gum, arabic cellulose, and FOS. Mice remained on these diets for 7 days before undergoing seizure testing. Now, the key findings were shown in Panel B, which presents seizure threshold measurements, specifically something known as CC50, or the electrical current required to induce seizures in 50% of mice. The results showed that the mice on the standard ketogenic diet with a 4:1 ratio already had a high seizure threshold. However, when extra fiber was added, CC50 increased even further. This means that boosting fiber levels significantly improves seizure resistance. Another important measure is latency to exploration, which tracks how quickly the mice resume normal activity after a seizure. The graph showed that mice on the fiber supplemented ketogenic diet took longer to return to exploratory behavior, which even further supports the idea that additional fiber enhances seizure protection. Now, if we go into panel D, which shows a heat map of metagenomic pathway changes, the addition of fiber increased pathways related to pre-QO biosynthesis and QO-SYN biosynthesis, both which have been linked to neuroprotection. At the same time, fiber supplementation reduced pathways involved in sucrose degradation and carbohydrate metabolism, which again aligns with the prior findings that suggest limiting these processes supports seizure resistance. Finally, the panel E reinforces these effects by comparing different fiber sources. The results show that the group receiving the fiber mix, including the extra inulin, gum, arabic, cellulose, and FOS, had the highest seizure threshold, outperforming even groups that received single fiber types such as gum arabic or oat fiber. And truly, the key takeaway from this is that not all fibers have the same effect. Some enhance seizure protection more than others. So, this study reveals a strong connection between dietary fiber, the gut microbiome, and seizure resistance. Earlier findings showed that ketogenic diets with higher fiber contents led to significant increases in seizure thresholds, meaning the mice were more resistant to seizures. On the other hand, the modified ketogenic diet, which lacks fiber, actually made mice more seizure prone. This suggests that fiber, not just fat content, play a crucial role in making the ketogenic diet effective. And the real true breakthrough came through when fiber was added back to the modified ketogenic diet. The results were impressive, seeing as mice that received fiber supplementation showed seizure thresholds even higher than the control group, proving that fiber restores and enhances seizure resistance. Now, the next question was whether adding even more fiber to an already fiber rich ketogenic diet would further improve outcomes. And as shown in Figure 4, the answer would be yes, extra fiber boosted seizure resistance beyond the standard ketogenic diet. But not all fibers work equally well, and the best results came from the specific fiber mix containing inulin, gum arabic, cellulose, and FOS, which were linked to beneficial gut microbiome changes. Altogether, these findings suggest that the optimizing fiber intake in ketogenic diets could be a powerful treatment for improving seizure control in epilepsy patients. And moving forward, clinical studies in humans will be essential to determine how best to apply these findings to epilepsy treatment. And to bring everything together, dietary fiber is not just a minor factor. It plays a key role in making ketogenic diets effective for seizure control. This study showed that fiber-deficient ketogenic diets, such as the modified ketogenic diet with a 3:1 fat to carbohydrate and protein ratio, actually lower seizure resistance. Meanwhile, fiber-rich ketogenic diets, such as the standard ketogenic diet and the medium-chain triglyceride-based ketogenic diets, enhance seizure resistance. But what's more important is that Adding fiber to a low fiber ketogenic diet doesn't just restore seizure protection, it actually enhances it beyond its baseline levels. And these effects are strongly tied to microbiome shifts, where fiber increases neuroprotective metabolic pathways while reducing carbohydrate degradation, which may be linked to seizure susceptibility. This fundamental shifts this fundamentally shifts the way we think about ketogenic diets, not just as high fat, low carbohydrate diets, but as therapies where fiber content is a key factor in seizure treatment. Now, why does this matter? First, these findings suggest that current ketogenic diet therapies for epilepsy may need a reformulation to include the right amount and type of fiber. Now, second, this research opens new doors for microbiome targeted dietary strategies. This could allow for personalized epilepsy treatment based on an individual's gut microbiome profile. This could also help with those who are unable to have the lifestyle of needing medication all the time. But there are still big unanswered questions, like which microbiome shifts are directly responsible for seizure protection, or are certain types of fibers more effective than others? And most importantly, can these findings translate to humans? And future research should focus on clinical trials testing fiber supplementation in ketogenic diets for epilepsy patients to truly determine whether these effects hold true beyond animal models. Now, one of the biggest strengths of the study is its comprehensive microbiome analysis. Researchers used both 16S rRNA sequencing to classify bacterial species and shotgun metagenomics to examine microbial functions. This allowed them to not only identify which bacteria were present, but also determine how they function metabolically. Another strength is the well controlled experimental design. Researchers carefully compared multiple diet groups and used rigorous amounts of statistical methods to ensure the results were meaningful and reliable. However, just like any research, there were limitations to the study. First, the study was conducted in mice, and while mice are useful for model for neurological research, human microbiomes are way more complex and may not respond the same as a mice model. Second, the study only lasted for 7 days, meaning we don't know the long term effects or if the microbiome would adapt over time. And finally, while we see clear microbiome changes, we still don't know which specific bacterial species or metabolites are directly responsible for seizure protection. So, further studies should aim to pinpoint exactly how fiber is working as at a mechanistic level. For me, this study challenges the way we traditionally think about ketogenic diets. We've always focused on fat to carbohydrate ratios, but this research truly shows that fiber, which is something that's often ignored in ketogenic diets discussions, could truly be a game changer in the field of epilepsy and therapy. Given that fiber already plays a role in metabolism, inflammation, gut brain signaling, it makes sense that it could also impact neurological health. And in conclusion, to put it simply, this study changes how we think about ketogenic diets and their role in seizure protection. We now know that dietary fiber is not just a minor component, it's essential for maximizing the anti seizure effects of a ketogenic diet. Low fiber ketogenic diets, such as the modified ketogenic diet, actually reduce seizure resistance, while fiber rich ketogenic diets enhance it. And on top of that, it's not just fiber that was beneficial. It was also the addition of more fiber that was equally as beneficial. It Adding fiber to a low fiber ketogenic diet doesn't just restore seizure protection, it enhanced it beyond baseline levels. So this effect is closely tied to gut microbiome shifts and truly reinforces the growing connection between diet, gut health, and brain function. If you look at these findings, there's a broad impact on epilepsy treatment and beyond. And these implications go even further. So, understanding the relationship between fiber, the microbiome, and neurological health could truly open new doors for treating other brain disorders such as Parkinson's disease and Alzheimer's disease, where gut brain interactions also play a key role. This research highlighted a simple but very powerful concept that what we eat shapes how our brain functions, and dietary fiber may be a key player in neurological health. Now, before we wrap up, here are a few key questions to think about. If fiber can enhance seizure resistance through microbiome-driven mechanisms, does this suggest a paradigm shift in how we approach dietary therapies for neurological disorders? What does this study reveal about the gut-brain axis, and could microbiome modulation be an alternative approach to neurological disease treatment? The study shows that not all fiber types have the same effect on seizure resistance. Now, how should future research determine the optimal fiber composition for neurological health? Could fiber alone, independent of ketosis, provide seizure protection, or is the interaction between fiber and the ketogenic diet essential? And could the findings of the study extend beyond epilepsy to other neurological disorders influenced by the gut microbiome? Now, here are my resources, and thank you so much for listening and paying attention to this presentation.